would now like to ask the next speaker, which is Edda Schulz from Institut Curie in Paris. And she will speak about two positive feedback loops, control effector function and memory induction in type 1 T-helper lymphocytes.
Does it work? Okay, first of all, I want to thank, of course, the MTZ Foundation for the prize and then the opportunity to speak here today. So, I will speak about my PhD work, which I did within a collaboration between the Humboldt University, where I worked in the group of Thomas Höfer and a lot with Luca Mariano on the theoretical parts, and the Deutschen Rheuma-Forschungszentrum, also in Berlin, where I performed all the experiments in the group of Andreas Rackko. So I worked on two positive feedback loops that control effector function and memory induction in type 1 T-helper lymphocytes. So T-helper lymphocytes are part of the mammalian adaptive immune system and play a crucial role in immunity. So what you see here on the left part is the primary infection with a pathogen, and the T cells that are specific for that pathogen now only respond with such a delay, as you see here, before they clear the infection. So but some of these T cells can now survive in the body for a long time, the so-called memory cells, and during a secondary infection with the same pathogen, these memory cells now can respond much faster and also much more specific, which is one of the reasons for immunity. So the reason why they respond so fast and specific is a differentiation process that they undergo during the primary infection, and which depends on the kind of pathogen that they encounter. So, for example, in response to intracellular pathogens, the immune system produces the differentiation signal interleukin 12, which together with the specific antigen signal induces differentiation of naive T-helper cell to Th1 cells. Now the Th1 cells produce cytokines such as interferon gamma, which mediate the effector functions. Now the differentiated cells, they essentially know already which is the right response for that pathogen. So they have like the memory that they can now produce interferon gamma in response to the antigen signal alone, which is the reason for this fast specific response. So my project, I tried to understand the gene regulatory network that controls this differentiation process. Therefore, I started by selecting a set of genes that were already known to play an important role in Th1 cells, which are shown here. So there's this transcription factor T-that, which is the so-called master regulator of Th1 cells, because when you overexpress it, even in Th2 cells, they will convert to Th1 cells. Then there is the receptor for interleukin 12, the differentiation signal, and this receptor is not yet expressed in the naive cell and must first be upregulated so that the cells at all can respond to IL-12. And third, there is interferon gamma, which I already mentioned because it mediates the effector functions, but also it functions in such an autocrine manner and is important during the differentiation process. So when I started by sort of constructing this network by what was known from the literature already about the regulation of these three genes. But already then there was a problem with this network, because if this was true, you could just put interferon gamma on the cells, which would upregulate T-that, which would drive Th1 differentiation. But it was known for long already that the main crucial signal here is interleukin 12. So something seemed to be missing in the network. So that's why the sort of central question I tried to answer was then, why can IL-12 but not interferon gamma drive Th1 differentiation? And to answer this, I proceeded in two steps, where first I asked what is missing in this network, and then how does this network control Th1 differentiation. So to complete the structure of the network, I started with developing a mathematical model describing this network based on differential equations, and then compared model simulations with experimental measurements. For the experiments, I isolated naive T-helper cells from mice, which were then differentiated in-cell culture by adding an antigen stimulus, which was only present for the first 2 days of culture, together with the differentiation signal interleukin 12. And then at different time points, I isolated mRNA and quantified the expression of the genes I was interested in. So this is what the expression kinetics that I found for the transcription factor T-Helfer. As such, it shows such a biphasic expression profile with one peak after 1 day of stimulation, but then a second upregulation after 4 to 5 days. So then I tested whether this initial literature-based model could account for this behavior. But as you see here, in the dotted line, it can explain, well, the first peak, but it cannot explain T-Helfer expression in the late phase of differentiation. And this is because here in this model, the antigen signal is required for T-Helfer expression. So since the antigen is removed after 2 days, there can be no T-Helfer expression in the late phase. So to understand which signals control T-Helfer expression in the late phase, now I systematically blocked regular pathways in this network and then measured how this would affect the T-Helfer expression kinetics. So first, I blocked interferon gamma signaling by using interferon gamma receptor deficient cells. And you see that now the first peak disappears, so it seems to really require interferon gamma signaling. But in the late phase, T-Helfer is still upregulated, so there seems to be another pathway of T-Helfer regulation that is independent of interferon gamma. So to understand this, next I blocked IL-12 signaling just by culturing the cells without IL-12 in the medium. And you see now the opposite effect. So now the first peak of T-Helfer is unchanged essentially, but in the late phase the levels are strongly reduced, suggesting that the first peak is independent of IL-12, but in the late phase IL-12 is required for inducing T-Helfer. So, and if I block both of these pathways, there's no T-Helfer expression. So that's why I integrated a new regulatory link in this network, assuming that IL-12 can directly induce T-Helfer, and tested again whether this now could account for this biphasic behavior. But now the new model can explain T-Helfer expression in the late